chairman of the session, moderators, my fellow colleagues are present. Assalamu alaikum. I'm Abdullahi Abubakar Lema by name. I'm a PhD research student at University Sultan Zainal Abidin, Malaysia. I'm here to present a conference paper titled Evaluation of Biochemical Response during Post-Harvest Deterioration of Hibiscus sabdariffa, Roselle calyx. My introduction will be based on the following content. My presentation will be based on the following content: introduction, objectives, material and method, result and discussion, conclusion. My way of introduction: Roselle calyx, Hibiscus sabdariffa, belongs to the family Malvaceae, which can be found in many tropical and subtropical countries around the world. Roselle have been widely used in medicine, foods, as color pigment, and many more. The calyx are traditionally prepared as beverages because they are rich in anthocyanin, which have antioxidant. The antioxidant properties of Roselle are nine times higher than that of the vitamin C, which is the standard non-antioxidant. Nowadays, people have recognized that food not only satisfy hunger, but also provide a numerous health benefits, including anti-cancer, anti-diabetes, anti-diuretic, antioxidant, anti-hypertensive, and many other medicinal benefits. Anthocyanin is a natural water-soluble pigment which is among the derivatives of flavonoids, and there are 6 non-anthocyanidins, namely pelargonidine, delphinidine, peonidine, pentinidine, malvidine, and cyanidine. And among these 6 anthocyanidins, the most prevalent found in Hibiscus sabdariffa are delphinidine and cyanidine. Roselle calyx is perishable. Thus, drying the calyx is challenging due to poor post-harvest deterioration and instability issue of anthocyanin. A lot of companies, for example, in Malaysia, over 18 companies are involved in marketing and processing of Hibiscus sabdariffa, and most of these processing companies prepared fresh calyx, but due to the prepared fresh calyx, but in terms of processing, in terms of transportation to the companies, the calyx deteriorated early. Deterioration can increase the production of free radicals, such as hydroxyl radicals, superoxide, and hydrogen peroxide. Under this condition, plant genes modulate flavonoid biosynthesis, including the expression of anthocyanin during citrus exposure. And furthermore, anthocyanin has been known to protect plant from oxidative stress by donating hydrogen atom. Research into molecular and biochemical mechanisms of PPD revealed that reactive oxygen species formation is among the first reaction, and several related genes and their expression have been also documented. Enzymes can be used to study physiological and biochemical changes in plant products that have been stored. Objectives of this study are to determine enzymatic and non-enzymatic antioxidant activities during deterioration of cell calyx, to explore the potential of anthocyanin in activating antioxidant in activating enzymatic response during post-harvest deterioration, to determine the novel post-harvest deterioration biomarker in Roselle calyx, material and method. The freshly harvested calyx was the freshly harvested calyx will be washed and cleaned with tissue paper and put inside a clean plastic bag at room temperature. The biochemical response or biochemical changes during deterioration at different time interval was monitored. The non-enzymatic antioxidant was determined. Total phenolic content was determined using valence equal to method. Total flavonoid content was determined using aluminum chloride. Total anthocyanin content was also determined using pH differential method, and percentage radical scavenging activity of DPPH was determined. The hydrogen peroxide concentration was determined, and enzymatic antioxidants such as catalase, ascorbate peroxidase, and peroxidase was also determined in these studies. And lastly, polyphenol oxidase and phenylalanine ammonia layers was determined in this research. Result and discussion. This Figure 1 shows calyx fresh and deteriorated uh, calyx, and the calyx usually deteriorated usually completely deteriorated uh, from 5 to 7 days after post-harvest deterioration. This is the result for total phenolic content and total flavonoid, which were all expressed, but later decrease at later stage of deterioration. This finding is supported by Topman and Rahman et al. Total anthocyanin content was also expressed, and uh, these are the delphinidine and cyanidine, and this delphinidine and cyanidine are the most prevalent anthocyanidins, and were also expressed during deterioration. This figure shows the schematic presentation of role of anthocyanin in response to citrus during post-harvest deterioration of Roselle calyx, which shows a coordinated effort uh, the anthocyanin have in activating antioxidant enzymes, which responds to hydrogen peroxide generated, which responds to hydrogen peroxide generated as a result of deterioration, and also anthocyanins themselves, they are antioxidant, and also the phenylalanine ammonia lyase was expressed, which is the first permitted enzymes in flavonoid biosynthesis pathway and anthocyanin biosynthesis pathway. The percentage radical scavenging activity was uh, shown. It has significant difference, even though at 5 days the 5 days decrease due to the deterioration. The decrease also in 5 days hydrogen peroxide was due to the antioxidant enzymes present, and this finding is supported by Bullock et al. The antioxidant enzymes catalyze peroxidase was expressed in this finding, and this finding is supported by the finding of Tecros and Liu et al. 2021. The expression polyphenol oxidase, which is the enzyme responsible for browning of fruit during deterioration, was also expressed in these findings, and phenylalanine ammonia lyase was also expressed during deterioration of Roselle calyx. This finding is supported by Banta et al. 2015. In conclusion, in this study, we determined both enzymatic and non-enzymatic antioxidants during Roselle calyx degradation process. The Roselle and the role of anthocyanin in activating antioxidant enzymes and related was explained during post-harvest deterioration of Roselle calyx. Uh, this could serve as a deterioration biomarker and help in achieving food security and reduce food loss due to post-harvest deterioration. Furthermore, polyphenol oxidase and phenylalanine ammonia lyase could also be potential deterioration biomarker due to their expression during Roselle. From this study, we have reported for the first time the biochemical changes during post-harvest deterioration and potential deterioration biomarkers in Roselle calyx to the best of our knowledge. And from our findings, the study to enhance the anthocyanin stability using CRISPR and other molecular techniques is recommended. These are some of the important references Thank you for the listening.